Welcome to Ever Wondered. This week we're looking at the newly discovered creatures of our oceans and trying to unravel the mysteries of the deep. This journey takes us to the farthest depths. We're meeting the scientists that are discovering new species by the dozen, investigating their carnivorous tendencies and trying to find out how they can be used to save lives. First, I want to find out how much in our oceans remains undiscovered. So I'm off to NIWA's vast invertebrates collection to meet Dennis Gordon, a leading expert in New Zealand's biodiversity. So Dennis, does New Zealand have a lot of biodiversity?
New Zealand doesn't do bad on a global comparative basis, yeah. We have more than 17,000 species that we know about. And in fact, the numbers that we can expect should be tens of thousands more than that.
Right, so there's a lot of things which are undiscovered.
Absolutely. In fact, we cannot keep up with the rate of discovery in New Zealand. In fact, we're finding new species faster than we can name them and describe them, yeah.
The main reason that so much remains undiscovered is that New Zealand has a huge exclusive economic zone. This extends for 200 nautical miles from any landmass, including our many tiny islands. In this massive area of seafloor is a massive range of habitats, from the 10-kilometre deep Kermadec Trench to underwater mountain ranges and vast plateaus. Just like on land, the diversity of these habitats is matched by the diversity of the residents. Ah, that thing's rad. What's this?
Oh, that's part of a giant bubblegum coral. It's the largest seafloor organism on the planet.
Is it? How big does it grow to?
It gets up to 7 metres, and so far as we know, it can live up to probably a maximum of 500 years. 500 years. What are these things on it? Why are they? It's covered in brittle stars. So, in fact, think of a bubblegum coral tree as like a tree on land, in which you have birds and all sorts of other things.
So this is kind of like the cowrie of the sea.
That's a fantastic analogy. This really is the cowrie of the ocean.
Cool. That looks alien.
Yes, a giant marine worm. This is from the Ross Sea. And a lot of Antarctic animals tend to get quite large, probably as a consequence of their slow metabolism, and they're able to live for a very long period.
Really is an alien world, isn't it?
Very alien.
The invertebrates collection contains between 300 and 500,000 samples, many of which are waiting patiently to be studied. This biodiversity, why is it important to us?
It's important to New Zealand because 50% of our marine life is found only in New Zealand. So from a global perspective, we should conserve that for the interest of the world, basically. We really don't know enough about our oceanic resource. We don't know how many species we have. I mean, I said we have 17,000 species that we know about, but we expect tens of thousands of additional species out there that are still undiscovered. The value is very high, both from the point of view of scientific interest, but also from the point of view of this constitutes a natural resource for New Zealand.
Right, so fishing.
Fishing would be the obvious one. A healthy ocean and healthier biodiversity means a healthier fishery.
One of the reasons that so much of our ocean remains unexplored is the difficulty of accessing it. But for ecologist Ashley Roden, the lure of discovering new species is too great. Crushing pressure, near freezing temperatures, and total darkness are just some of the problems he faces as he ventures into the deep.
Most of the projects that I work on involve the deep sea itself, so generally at depths deeper than 200 to 500 metres, down in fact to the deepest depths possible in New Zealand, down to 10 kilometres in the Kermadec Trench. Generally, every time you go out there with a grab or a trawl, the chances are you're going to pick up some species which has never been seen before by science.
This might seem surprising because the deep sea floor appears to be such a barren landscape.
The deeper you go in the ocean, generally the productivity drops off. You can't get food down from the surface waters. As it's drifting down to the deepest depths, it gets broken up by bacteria and other organisms actually in the water column. So by the time you get down to 10 kilometers, the quantity and the quality of the food is much reduced, and therefore it's going to support a lot less animals.
But Ashley is studying the exceptions to this. There are some areas where thriving ecosystems are powered not by the energy of the sun, but by chemicals from deep within the earth. At undersea volcanoes, superheated chemical-rich fluid is emitted, providing the food for microbes that form the first link in a completely different food chain.
This is amazing.
It is exciting when you see it because it's quite a different from something just 100 meters to the left where you might get a coral here or a snail there.
Why is there so much life there?
That's right, it's a very toxic environment. There's very high temperatures, there's all sorts of, if you like, poisonous chemicals, where they're poisonous to the majority of life. But there are some organisms which can actually live in this amazing environment. So bacteria, microorganisms can survive, and they use sulfides and other chemicals which are emitted through this fluid to provide energy for themselves. And they're used by another suite of animals. So you build up a whole load of organisms which are essentially going all the way back to chemicals which are coming out of the earth.
Such an unusual energy source gives rise to the evolution of a very different community.
Drifting back down. Big one, shove it.
So the number of species that can live in these toxic environments is usually quite low, so species diversity is low, but abundance and biomass is huge. And that's quite something to see when you go in the deep sea, because normally that's not the case. So when you arrive at these oases on the sea floor, what you see is lots and lots and lots of organisms. You see swarms of shrimp, you see fields of swaying stalk barnacles, you see many, many mussels and big mussel patches, mussel beds. And it's quite amazing to see that sort of huge abundance of life.
Ashley has discovered many new species at these isolated communities, like these clams. And this coral-like creature here is a bryozoa, the first ever found at event, and named after the man who discovered it, Parachnoidea rhodoni.
How many new species have you discovered?
Oh, I think it's not possible to say how many. I mean, the number of species which have come from samples which I've been part of would go into the hundreds, possibly the thousands.
That must be pretty exciting.
It is exciting, but at the same time it's pretty daunting because it means that you're building up this backlog of species which still need to be described by people, and we don't have that many people to describe them.
One of these specialists is the sponge lady, Michelle Kelly of Niwa in Auckland. Once intrepid biologists like Ashley Rowden have discovered what they think to be a new species of marine life, they need to find somebody with the expertise to examine it and figure out exactly what it is. This is called taxonomy, which is the scientific practice of classification and naming. And when it comes to sea sponges, in New Zealand, Michelle Kelly is the only person for the job.
So what is it about sponges that's really interesting? Well, the first thing is that they are animals, they're not plants. A lot of people think they're plants. They're really just an accumulation of about 17 different cell types. They have no eyes, no mouths, no stomach, none of the usual things that we expect in animals. But to me, the most fascinating thing was that if you were to squeeze a sponge into a petri dish, it would release the cells. And those cells would be able to recognise each other as from the same sponge, and therefore they would then kind of clump together and regrow the sponge.
It sounds a bit like a monster from a science fiction movie. So it's no wonder that Michelle has made her small lab in Auckland into the sponge taxonomy epicentre of New Zealand.
Yeah, don't be too worried about it, it's not going to break.
Looks fragile.
Right.
As you can see from Michelle's collection, sea sponges come in all shapes and sizes, and most are not at all the soft, bouncy things that I expected.
This is a glass sponge. Originally that would have been like a beautiful tulip.
This is the top.
This is the top, and this would have been buried in the sediment. It's incredible.
It looks like shell.
Yeah, it does. It's all just silica, that's big silica strands that have been cemented together. This is a traditional bath sponge from the Caribbean. So if we were to put that in water, it would just go into a beautiful soft mass, and then you go and scrub yourself with it. Oh. You wouldn't recognise this in life, because this is only the skeleton. So in this would have been all the cells.
So when you harvest it, it doesn't look like this at all?
No. It looks like probably like a black fleshy sort of thing,
Okay.
a blob.
The work of taxonomists like Michelle form the basis of all biological study and could be referred to for hundreds of years. She examines the skeleton of a new sponge to look for similarities with other sponges so that she can figure out where it lives in the larger sponge family. Of particular interest is a skeletal element called the spicules. These provide structural support and also deter predators. They are useful to Michelle because of their seemingly infinite variations.
I mean, look at this little thing. It's got little trident-like ends, so they not only do they go in, but they stay in. They're like little barbs.
Cheeky little
So I'd
bugger.
be careful if I were you, yes.
These examinations often throw up unexpected discoveries. The one that's rocking the sponge world at the moment is that not all sponges are herbivores, as was previously thought.
A normal sponge filter feeds. It has a layer of cells which are flagellated, so every cell has a little tail on it. And what they do is they draw a current of water in through the outside of the sponge. Those cells trap food particles, and then the cells pass those bits of food back to the other cells.
But Michelle discovered a new breed, a carnivorous sponge found just north of the Chatham Rise. So unusual and fascinating was this that the International Institute for Species Exploration named it one of the top 10 new species of 2010.
Can I pull it out
Yeah,
here?
pull it out, yep. His name's Chondrocladia malleoderma turbiformis.
And this is carnivorous.
That's a carnivorous sponge, yes. It eats little tiny shrimps in the deep ocean. What it does is that the little shrimp is just sort of minding its own business, sort of, tromping along like this. And then it maybe tries to sit on one of those or says, oh, what are you?
Nice place to rest.
Yeah, whatever.
Yeah, okay.
And then it just gets all tangled up and caught up, and then because the thing's wriggling around, it keeps catching more and more. It's a bit like Velcro, when you're trying to pull Velcro off? Well, it can't escape, and so gradually the arms sort of go like this. Sorry, it's very dramatic, but eventually the cells just sort of join up with their other mates and just sort of start, enveloping it like
And
this.
then it's just gone.
And then it's just gone. Yeah, it's subsumed.
So that must have been exciting to see a new way that an animal feeds.
Well, it is, and especially because sponges simply don't eat this way.
Right.
We were actually questioned whether they were sponges.
There have now been many carnivorous sponges discovered in New Zealand waters, displaying extremely sinister spicules that they use to ensnare their prey.
Is this just for science? Like, why should the public care about the naming and identification of sponges?
We need to know what's in our waters before we start mucking around with them.
Better understanding means better conservation.
Exactly. But there's a lot of other reasons why we need to know the different sorts of sponges, and that's because they have lots of very interesting chemical compounds in them.
What does
And
the sponge use those for?
That's a good question. I think some of them use them to defend themselves, protect them from predation. See, they don't move, they're just sitting there, so how do they protect themselves? So they seem to be able to produce these chemicals, and that will, like the fish will take a bite and just spit it out. They don't
Just
like
yuck,
it.
don't want it.
Don't want it.
This means that sponges contain toxins, and new sponges are likely to contain toxins with a completely new way of acting on the human body. Medical science is looking at these toxins in the search for this generation's holy grail, a cure for cancer.
We use submarines to find these amazing rock sponges that lived on the continental shelf of the Bahamas. And some of those sponges contained chemicals which are currently in clinical trial for the treatment of cervical cancer in human females. Now, how does that work?
This is the story of how a discovery in 1982 by two Kiwi scientists eventually led to the creation of an anti-cancer drug that was released in 2010. But I'm not off to a university chemistry lab this time, because the process has taken so long that the scientists involved are now retired.
Oh yeah, Murray.
Murray, how you doing, John? Murray Munro has kindly agreed to host me and his colleague John Blunt so they can tell their story that began with the discovery of a very special sponge.
We called it yellow slimy, which tells you quite a lot about it because it was bright yellow and it was also very, very slimy.
At the time we collected it, we had no knowledge that it was of any interest. It wasn't until we got that material back into the laboratory and did the extraction and did the bioassay, determined, hey, that sponge was had something pretty potent in it.
The bioassay is the first crude step down the long and winding road of testing a new drug. It allows scientists to screen extracts from many different organisms quickly.
So we get an extract and then apply a drop of that to a plate which has growing on it mouse leukemia tumour cells and observe whether that drop of extract was able to stop the growth or kill the cells.
It had a test upon control against animals of 250%. Anything better than 120% is considered good, very good, interesting, and we had 250%. And so we went, wow. And so we just knew that this was incredibly powerful.
While the results were incredible, all they really prove is that the extract was very active against mouse leukemia cells in a petri dish. Not what impact they would have against human cancer, or what toxic effect they would have on the rest of the body.
We're chemists working in the chemistry department, and we can only go so far with the development of a compound. We do the initial biological assays to determine that something is of interest, but beyond that we have to give it to others who have all of the other biological assays and tests available to them to establish the viability of something as a potential drug candidate.
It takes hundreds of millions of dollars and many years of research and testing to bring a drug to market. But Murray and John's published findings soon attracted the attention of the most powerful cancer research body on the planet.
The National Cancer Institute became interested because they wanted a large supply of this material. And so they commissioned us to make a large-scale collection. But first of all, we established that there was about 300 tonnes down there, and we were then given permission to collect 1 tonne.
This was a huge operation and quite unprecedented in this country. Eventually, from this 1 tonne of material, Murray and John extracted just 250 milligrams of the compound now known as helicondrin B. But this was enough for the National Cancer Institute to begin testing. Results were very promising, but it was clear that the supply of sponges was not plentiful enough to continue.
This is the real dilemma for anything which is coming from a natural origin. You've got to get the supply somewhere along the line. And coming from a limited resource, 300 tonnes in New Zealand, it's not enough. So what can you do next? And the only answer to that is to look at the possibility for synthetic options.
Creating a synthetic form of the natural compound is a key step before going into full-scale trials. This is a 3D model of a drug molecule, and as you can see, it's very complex. If you want to go into clinical trials for a drug, you need to make kilos of this because you can't buy it. You can't even buy large sections of it. What you need to do is make it from fundamental chemical building blocks. To make it, I first need to look at the compound and see which parts I recognise. So as a chemist, I know that I can make this section from very cheap, readily available chemicals, namely acetic acid and ethanol. I put these two into a very simple chemical reaction. I lose water as a byproduct, and I end up with what's called an ester. So this is this section here. To make the entire molecule, I do that many times over using a lot of different chemical reactions. But the more complex a compound is, the more steps you take, the longer it takes, the more waste you have, and the more expensive it is. But what you can do is have a look at the drug molecule and identify which part is actually having the effect inside the body. This means you can get rid of this bit here, which isn't having a direct effect. Neither is this part doing the business, or this part. So what we end up with is a drug molecule that has the same effect, but it's much simpler, which means there's less steps, there's less waste, and it's cheaper. And this is called an analogue. The analogue for helicondrin B was named eribulin and was first synthesized in 1992. Since then, Murray and John have been observing the progress of their discovery from afar.
We knew that we would always be at the front end of the discovery process. Someone else would then have to take that stage further to clinical development and eventual drug release.
After going through the rigorous process of animal and then eventually human trials, eribulin was finally given the OK by the Federal Drugs Administration in the US and became available for use against breast cancer in 2010. It is now tipped to become a $1 billion a year product. 28 years after Murray and John first pulled yellow slimy up from the depths of the Kaikoura Trench.
Well, when we got the news that this was going onto the market, we were pretty pleased.
Smiles.
No, we were.
There was always that little sneaking suspicion it would be wonderful to get something which would go all the way through to an anti-cancer drug. And to have actually then seen this work being released is a big thrill.
Murray and John are now retired, but they have left behind them a legacy that will make a permanent mark on marine natural products research in this country and across the globe.
I think one of the really nice things which has come out of this is we started this up in the mid-1970s, just the two of us, and then it's spread out with our former students.
And to see these people leave our care, if you put it that way, and go out into the world and make their own way, and some of them are doing very well. You know, there's rewards for us for having done this work as well in other ways, quite apart from, I suppose, international recognition for the work that we've done.
The journey of science is a long one. From all the work that goes into discovering a new organism, through figuring out what on earth it is, and perhaps even finding an amazing chemical property that can be harnessed for the benefit of humanity. But the biggest lesson from this journey is that we know so little. New Zealand's deep water still holds an unfathomable number of mysteries, waiting to be unravelled by our scientists.